And thank you for organizing this amazing meeting. Thank you for giving me the opportunity to share some ideas with you. What I'm going to do today is talk about some symmetries, what I think are profound symmetries between neuroscience and developmental biology. And if you want to see any of the details, the software, the data sets, everything is at this site. And then here are some kind of personal thoughts about what I think all this stuff means. So I'm going to frame today's talk around this question of how does it know? Because when we look at biology, and in particular developmental biology, that's one of the first things that strikes people is how do the cells and tissues and everything else know what to do? So I'm going to give a few main points. First of all, I'm going to claim that biomedicine and bioengineering boil down to finding out the informational structures of the material of life. That is, how does it know what to do in various circumstances, what kind of mistakes it makes and why, how do we alter decision-making towards specific set points that living material is very good at doing, and for biomedicine, and engineering in particular, how do we change these outcomes with the least effort possible? And I'm going to make three basic claims: that bioelectricity is a kind of cognitive glue that binds individual cells and other structures towards larger scale purpose. I'm going to claim that bioelectricity is also a kind of memory medium, which basically stores patterns towards which cells and tissues operate, and that we can now reset those patterns. And then towards the end, I'm going to show you some new bioengineering, which I think is a great opportunity for bioelectricity, but it hasn't really taken off yet, but I think it will. So the most obvious thing that anybody first asks when they see this process of embryonic development, where we all start life as a single cell, and eventually we become one of these amazing things. Basically, the first question arises: how do the cells know what to do? They all have the same genome. The stem cells and everything else in your hand and in your foot are the same. But why does your hand not look like your foot? How does it know when to stop? All of these things. And so this question of how do they know what to make, I'm going to emphasize the idea that the mechanisms of knowing what to build and how are actually homologous, meaning both in terms of their mechanisms, their evolutionary mechanisms, and their algorithms, to those that underlie knowing in familiar contexts, meaning behavior, brain-driven behavior. These are some of the exact same mechanisms that operate in those two spaces. And the first clue we get to this is this interesting point that chemistry doesn't make mistakes. Chemistry just does what it does, and the laws of chemistry sort of roll forwards, and that's all. But morphogenesis absolutely can make mistakes. And so something very interesting happens in this transition, where you go from chemistry and physics to a morphogenetic system that has goals and the ability to fail to meet them, and then to various cognitive and behavioral goals that also can be met or not. And this kind of thing was well appreciated by Alan Turing. He was a guy who was very interested in intelligence, broadly defined in different embodiments for intelligence, machine learning and machine thinking and things like that. But he also, towards the end of his life, he wrote this amazing paper, The Chemical Basis of Morphogenesis. And we might wonder, why would somebody that was interested in computation and intelligence be thinking about chemicals during morphogenesis? But I think he actually saw this profound symmetry, that the story of the autopoiesis of minds is basically the same story as the autopoiesis of the body, that we have to understand how this works. And we encounter the amazing aspects of this self-constructing material very early on in development. This is a single cell. You can see it's very active. It has lots of competencies in its own little cognitive light cone. This is a free living being called the Planaria, but we're all made of cells like this. And for that reason, we are all a collective intelligence. We are all made of competent little subunits. In fact, during embryonic development, when we look at an embryo and we say there is one embryo, what are we counting? What is there one of? There might be hundreds of thousands or millions of cells. What is there one of that we're counting? Well, what we're counting is alignment. We're counting commitment of all the cells to the same journey in the anatomical space. What makes it an embryo as opposed to a pile of cells is that they're all committed to the same homeostatic process that's going to get them to a particular region of anatomical space. They're all going to make this thing. And in fact, actually, what you can do is you can make little cuts in this plastoderm, and here are some in an avian embryo that I made many years ago. And when you do this, each of these little islands, for the time that it doesn't feel the presence of the others, it self-organizes into its own embryo. And so from this excitable medium of this plastiderm, you might get 1, 2, 0, or up to half a dozen or more individuals. So the question of how many individuals are here is not an obvious question. It is not set by the genetics. It is solved in real time by processes of alignment, by this, and this is, I'm going to mention this again and again, this notion of cognitive glue, these mechanisms that enable individual pieces to align in some kind of problem space, in this case, the anatomical space, to have a shared vision of what it is that they're going to build. And bioelectricity is a really important, it's not the only mechanism, but it's a really important cognitive mechanism that allows wholes to form. By the way, of course, in cognitive science, we already know this is the case because electrophysiology is what makes us more than a collection of neurons. It is what allows us to have memories, preferences, goals, and so on that our individual neurons don't have. And there too, you have this exact same kind of question about how many individuals are within a certain amount of real estate, because we have split brain patients and dissociative identity disorders and things like this. So this is, again, very, very parallel. So we need to understand how all these kinds of decisions are made. And the standard story is the genome. In fact, more than that, the standard story that we're told is this kind of open loop process that leans on notions of complexity and emergence. That basically there are these gene regulatory networks, they make proteins. Some of the proteins do things, they diffuse, or they're sticky, or they have enzymatic activity. So there's a bunch of physics that goes on in parallel. And then this magical process of emergence happens. And we know this is true. If you have lots of simple rules that you execute in parallel, often the outcome is quite complex. And so the standard story is this kind of an open loop feed forward emergence of complexity. But there's a significant distance between what's actually in the genome and the thing that we really want to understand and control. So this is a cross-section through a human torso. You can see the incredible complexity. Everything is in the right place, the right size, the right shape, next to the right neighbors. But what's actually in the genome, of course, is not anything about this. What you see in the genome is information about protein structure and some other information about when and how these proteins become expressed. It doesn't say anything directly about the size, the shape, the symmetry of the body or anything like that. So we still have this distance that we need to bridge between what's actually the hardware specification that is in the genome and the kind of physiological software that enables cells to know what to do and when to stop. And from that, we can infer how do we convince cells to repair things that are missing or damaged. And then, of course, as I'll show you at the end of this talk, more so, what can we get them to build other than the default morphology? So one thing to realize is that the genomic information is actually really insufficient for a broad understanding of shape. So just as a simple example, axolotl larvae have little forelegs. Frog tadpoles do not. And so in our group, we make something called a frogolotl, which is a kind of combination of frog and axolotl. And I could ask a simple question. We have the axolotl genome. It's been sequenced. We have the frog genome. It's been sequenced. So we have all of that. Can you tell me if a frogolotl is going to have legs or not? And the answer is we can't. We have no idea. And we also, and if it does have legs, whether those legs will contain frog cells or whether they will be made strictly of axolotl cells, we don't know that either. Actually, to be fair, we couldn't even predict the shape of either of these things from their genome, other than comparing it to the genomes of other animals that we do know what their shape is. So our ability to predict either static shape or these kind of novel cases is actually quite poor. And the other thing that's is really not readily discernible from genomic information are the properties of dynamic robustness. So, for example, this axolotl will regenerate its eyes, its jaws, its limbs, its spinal cord, portions of the brain and heart. And if you amputate anywhere along this axis, the cells will grow exactly what they need, and then they stop. That's the most magical thing about regeneration, is that it knows when to stop. When does it stop? It stops when the correct structure has been produced. Okay, so it's a kind of, you can see already, it's a kind of homeostatic process where the system can tell it's been deviated, and it will do what it needs to do to get back to where it needs to go. So all of these kinds of things, the capability of these systems are not readily predictable. Not only that, not only can these systems get back to where they need to be after injury, after some kind of external perturbation that damages a body part, but there's some really incredible problem-solving competencies within this material. So this is one of my favorites. If you make polyploid newts, meaning extra copies of the genetic material, what happens is that these kidney tubules, which usually have about 8 to 10 cells that work together to leave this lumen, the cells get bigger to accommodate the new genetic material. The newt stays the same size, so it uses fewer but larger cells to do exactly the same thing until the cells get truly gigantic, and then it'll use just one cell bent around itself, okay, and give you the same structure. Now, the ability to use different molecular mechanisms, so here cell-to-cell communication, here cytoskeletal bending, the ability to use different affordances in your toolkit to solve a problem you haven't seen before is basically a standard definition of intelligence. It's what's measured on all the IQ tests. What we have here is the ability of a newt, when it comes into the world, it doesn't know in advance, never mind the environment has uncertainties, but its own parts are unreliable. You don't know how many copies of your genome you're going to have. You don't know how big or how many cells you're going to have. And you have to do the job using different molecular mechanisms, right? So different affordances from your genome in ways to solve the problem. So again, all of this is completely not obvious from anything that we're going to get from the kind of typical molecular profiling. In fact, even below the single cell level, just the molecular pathways themselves, never mind the cells, never mind the brain, but just the molecular pathways by themselves have 6 different kinds of learning capacity, including Pavlovian conditioning. And so this is something else that we're doing in our group, is trying to take advantage of some of these properties for things like drug conditioning and so on. And the fact that the molecular pathways inside of cells can learn as well. So all of this forms this kind of amazing multi-scale competency architecture where the material has various capabilities at different levels, and all of the different levels have agendas and abilities to solve problems in different spaces, in gene expression space and physiological space and anatomical space. So ultimately, what we would like to do is this. We would like to have something I call an anatomical compiler. The idea is that someday you should be able to sit in front of a computer and draw the plant, animal, organ, biobot, whatever it is, in any shape and configuration that you want, okay? And the system should be able to then, if we properly understood how the material of life works, the system would compile this into a set of stimuli that could then be given to cells to get them to build exactly this, in this case, this three-dimensional flatworm, three-headed flatworm. The key here is that this is not something like a 3D printer, which is going to basically build as if it were Legos, build a whole structure. This is actually a communications device. It's a translator from the goals of the engineer to those of the cellular collective. And if we had something like this, all of this would go away, right? We could, if we knew how to communicate novel goals to groups of cells, all of this would become a non-issue. We're actually very far away from this. We don't have anything remotely like this. And you might wonder why, because molecular biology, genetics, biochemistry have been going very, very strong for many decades now. Why don't we have something like this? And partially it's because we've been really focused on this idea of the reliability of development. So this happens all the time, and it happens correctly almost all of the time. So you have eggs, and they give rise to a very specific thing. And we tend to think that, okay, well, this is what the genome encodes. This is what the genome is capable of doing. And we're still missing the deep lessons of both neuroscience and computer science, where a reprogrammable material is far more than its hardware specification. Because when you have something like this, and I purposely start off with a plant example, and we'll move into animals, what these cells are actually capable of is building things like this. This is called a gall. It's made of the actual cells of the plant. You would have no idea that the cells that reliably build this nice flat green structure, very stereotypical, we think that's what the genome does. We'd have no idea that they're actually capable of building something like this until a non-human bioengineer comes along, in this case, a wasp, that is able to prompt these cells with cues to build these incredible structures. And so who knows what else they're capable of? Probably the latent space is extremely large. But what we do know is that more advanced engineers produce more elaborate constructions. So bacteria make these kind of featureless blobs of, fungi do something similar. Nematodes are starting to do a little better. There's some kind of non-trivial structure happening here. Mites do the same. By the time you get to insects, you get these incredible constructs. So clearly the space of possibilities is far wider than the genomic default, than the thing that we see all the time. And what we would like to do, I mean, presumably it took that wasp millions of years to get to be able to do this. We would like to do it a lot faster. So in my group, in collaboration with Josh Bongard, we are building this kind of automated robot scientist platform that is basically making hypotheses about the laws of morphogenesis, actually providing stimuli to real cells in parallel, observing the morphogenetic outcomes, revising its hypotheses and going back. I mean, you'll recognize this as the typical cycle that we all do. In science, ideally, this will go a lot faster, so that we can actually start to have some control and understanding of this process. So where we are now is that the community is very good at this kind of information, figuring out the hardware, which proteins and RNAs interact with each other and so on. But what we would really like to do is this. And one kind of positive control that we can look at is what happened in computer science and information technology. This is what programming looked like in the 1940s and 50s. You had to physically interact with the hardware. She's sitting there rewiring the machine to get it to do something different. And this is what most of today's biomedicine and molecular medicine especially is all about. Genomic editing, pathway rewiring, protein engineering. It's all about the finer and finer control of the hardware of life. What we really need to understand is things like this. And so this was discovered in our group by Danny and Laura Vandenberg. And this amazing thing that yes, normally you have this stereotypical change of these tadpoles become frogs and they have to rearrange their face and move their various craniofacial organs. But this is not a hardwired process, because if you scramble all of them and you make these so-called Picasso frogs, where the eye's on the back of the head, the mouth is off to the side, everything is scrambled, you still get largely normal frogs, because all of these things will actually adapt to their novel starting conditions. They will move in novel paths. Sometimes they go too far and actually have to double back a little bit, but they still get their goals met. And so what you see is that the genetics is not giving you a piece of hardware that does the same thing every time. What it actually gives you is an error minimization scheme and a system that can recognize unexpected states and to take corrective action. And so that leads to a very obvious problem, which is how does it know what the right pattern is? If it's going to it's going to become a frog from the state or any of the other things I showed you, how does it know what the correct pattern, final goal is? So what we do in our group is augment this kind of conventional open loop with this homeostatic component, where actually deviation from this state, and this is something that typically open loop systems like cellular automata and so on don't do this. When you try to deviate the system from whatever that outcome was, it actually works really hard using all the mechanisms at its disposal. So of course the genetics, but also the physics, works really hard to try to get back there. And this is not just injury, but it's also mutations. It's also teratogens. It's also many different things. So what we would like to know then is how does this work? First of all, this is what, once you add this loop, for the first time, the notion of mistakes enters the picture. Because until you have a homeostatic loop towards a specific outcome that is willing to expend energy to get back to where it was, there's no reasonable definition of what a mistake is, right? Otherwise, it just sort of rolls forward and whatever happens, happens. But now you see that any situation that pulls it away from this state, and that might be a situation that's fixable or one that's not fixable, is now you can define this idea that how hard is the system willing to try to get back. How much stress is the system under, given that you've deviated from its goal state, and so on. And so what we need to understand now is, how does the system know what the set point is? Any homeostatic process has to have a set point, which is with respect to which it's measuring error. And so now we need to understand what does it actually mean to store a target state in cells, and what does it mean to try to reduce the delta from where you are now to where you are? How could cells possibly execute something like this? Well, we have a non-controversial example of that, and that, of course, is brain-based behavior. So in the brain, what we have is a bunch of hardware, which is constituted by cells in a network that communicate electrically. And on top of this hardware runs some very interesting physiology, which allows that network to do context-sensitive behaviors, to store memories, and do problem solving of various degrees of complexity, from simple habituation and sensitization all the way up to anticipation, planning, and many other things. And so what we know is that this system is using the remarkable properties of electrical networks and information processing in those networks to move your body through three-dimensional space. I mean, it does many other things now that we're humans and we play chess and do things like that. But fundamentally, it evolved to move you through three-dimensional space. And so we see all kinds of creatures doing all kinds of clever things, such as these crows that pick up cigarette butts and get a reward when they drop them off. And you ask, how does it know what to do? And so the commitment of neuroscience is that if we were to scan and read out this electrophysiology, we would be able to eventually decode it and extract the cognitive algorithms. We would know what is the animal thinking about, what are the memories it has, what are the goals, the preferences. We would like all of that cognitive stuff is encoded and implemented in the electrophysiology of the brain. And that's what these things think about. Now, it turns out that this amazing trick is, of course, not just about brains. This is evolutionarily very ancient. It evolved around the time of bacteria and then bacterial biofilms. And if you ask what those networks think about, I think the answer is they think about the movement of your body configuration in anatomical morphospace. Basically, I think what evolution did was pivot some ancient tricks that these electrical networks were doing to navigate anatomical space, and it basically sped them up quite a bit and then pivoted them into control of motion in three-dimensional space. And you can ask the same kind of questions. When Farinella grafted these tails to the flank, to the side of a salamander, they slowly remodeled, right? These tails slowly remodeled into limbs. And in fact, the tail tip of here, which these cells are in the correct local environment, they're tail tip cells sitting at the end of a tail, but they become fingers. This whole thing starts to remodel to better match the large scale pattern of what the body plan is supposed to be like. And you can ask exactly the same question: how does it know? How does it know what to do? It's basically a very parallel kind of system to what's done in neuroscience. And so we're trying to do exactly the same thing, to do this kind of neural decoding, except not in neurons, and to ask how are the decisions, the memories, the set points, and so on, how are these things encoded in the electrophysiology of somatic tissues? Okay, so same kind of question, using many of the same tools. And so here are some tools that were developed. So these are voltage sensitive reporter dyes. And so here's a movie that Danny Adams made years ago of a frog embryo during the time when all the cells are figuring out who's going to be left, who's going to be right, who's anterior, who's posterior. You can see all the conversations that these cells are having with each other. Here are some explanted cells, also some explanted amphibian cells in culture, making some decisions about who's going to stay within this group and who's going to leave. And you can see there's some important bioelectrical signals that Patrick McMillan in my group is analyzing. So we have the ability to use both dyes and genetically encoded reporters to read the electrical information in vivo. We do a lot of computer simulation, all the way from the molecular networks that give you those ion channels up through large-scale network properties of things like pattern completion, such as people study in connectionist computer science. We try to understand how artificial neural networks encode pattern memories and how they can be repaired and so on. And so here are some examples of what these patterns look like in vivo. This again is this famous electric face movie that Danny made, where you can see in a time lapse of this frog embryo putting its face together. Here's one snapshot from that video, where you could see basically a subtle pre-pattern that tells you here's where the animal's right eye is going to be, here's the mouth, the structures out to the sides, and so on. And not only is the bioelectrics critical for the cells in the structure to know what they're going to make, so it's a glue that coordinates the actions of individual cells towards a large-scale structure within one embryo. These kind of dynamics also work across scale. So, for example, here's an injury wave that propagates between individuals. So when when I poke this one, all of these find out about it. right? And so you can see here that it kind of, in effect, so this is Angela Tung's work, who showed that groups of embryos are actually much better at resisting teratogens than singletons, precisely because they communicate as a larger scale, a second tier collective intelligence. And again, you can see in these explanted cells that Patrick made, you can see the kind of two layers. You can see the slow bioelectrics that are happening, and then also here are the neurons, right? So we have the ability to observe all of this on different scales of time, on different scales of space. But then, better than just being, in fact, probably more important than just being able to observe these patterns, is the ability to modify them. You have to be able to do functional experiments and to insert information into the networks to know what that information is actually doing, and to be able to control outcomes. So in our group, we do not use applied fields or electrodes, no electromagnetics, no waves, no frequencies. What we do is manipulate the interface that cells are normally using to hack each other. That is, all the things I'm going to show you is not, they're not happening because we're so smart. They're happening because we're simply hijacking a system that already exists, by which these cells try to tell each other what to do and synthesize into a larger collective. And so what we basically do is we can control the connectivity of the cells via targeting gap junctions, or the actual voltage states of the individual cells. And we do this with optogenetics, or with drugs that open and close these different channels, and so that corresponds to synaptic or intrinsic plasticity in the case of neuroscience. So here are some things that we're doing. So in work with David Kaplan's group, we showed years ago that one thing you can do on a single cell level is control differentiation. So you could tell individual cells to be more stem-like or be differentiated depending on the voltage that you can control. But more importantly than individual cell state, because I think really the big impact of bioelectricity is going to be not at the single cell level, but actually in its collective forming properties, are the idea that when you connect cells, actually anything, but in this case cells, into a larger network, one of the things you're doing is scaling up their cognitive light cone. And what I mean by the cognitive light cone is simply the size of the biggest goals that they can pursue. So if we just sort of collapse space onto one axis and time onto the other, you can see that individual cells have little tiny goals. These are things like maintaining pH, maintaining metabolic state. They're all basically, they have a little bit of anticipation potential, a little bit of memory going back, but basically everything is in single cell organisms is concerned with maintaining the conditions at the level of a single cell. But groups of cells, such as tissues, organs, and whole embryos, they can have actually very large scale goals. They can start building things like this. So during development and during evolution, what actually happens is by joining together, the size of the goals that they're pursuing gets radically increased. So while individual cells only care about what's going on inside their own borders, these cells work really hard on this grandiose goal of maintaining a large structure. No individual cell knows what a finger is or how many fingers it's supposed to have, but the collective absolutely knows in a very functional sense, meaning that you can, if you know that they know this, then you will be able to predict that once you introduce damage, it will actually build the right kind of stuff, and that's when it will stop. Okay, so this system has a functional ability to get back to a goal state that is much larger than the tiny sort of scalars that individual cells pursue. But of course, this kind of thing has a failure mode, and that failure mode is cancer. So one of the things that happens when cells electrically disconnect from the collective is that they can no longer pursue these large grandiose set points. They basically become they basically roll back to their ancient unicellular lifestyle. And so here's a video of some glioblastoma cells that Juanita in our group studies. And there's been many papers looking at how these things are basically traveling back in their evolutionary history to occupy themselves with very small kinds of set points, and basically treating the rest of the body as external environment. What's happened is the border between self and world has shrunk. Now, this failure of this multicellularity does not require DNA damage. It doesn't require mutations. It can be triggered purely bioelectrically, because these networks that keep cells harnessed towards specific goals are in large part bioelectric. And so this is some work by Maria and Doug from our group, where we basically showed that if you interfere with the communication the electrical communication between melanocytes, these little pigment cells, and another population of a rare population of cells we called instructor cells, because they're the ones who keep these guys under control mostly. If you interfere with that, then these little melanocytes go crazy, and they acquire this hyper invasive morphology. They start to drop down and invade into the neural tube and the brain, and here in the blood vessels, you can see. I mean, this has a lot of the anatomical and molecular markers of metastatic melanoma. There is no primary tumor here. All of the melanocytes go crazy and do this, and there also is no genetic damage. There are no carcinogens here. There are no oncogenes, but they will turn on a bunch of markers that are associated with the metastatic melanoma. That's all done just by disrupting the coordination, the electrical coordination between cells. Better than inducing this kind of transform behavior, you can actually suppress it. And this is something that Brooke Chernet showed in our group, where you can first of all, if you do inject human oncogenes, so nasty things like KRAL and dominant negative P53 and so on, you will get tumors. But if you also co-inject a channel that forces the cells into the appropriate bioelectrical state and keeps them in coupled to their neighbors, then you won't get a tumor in a good chunk of the cases. This is the same animal, and so the oncoprotein is blazingly expressed. You can see it all over the place, right? It's marked with this fluorescence, but there's no tumor, because it isn't the genetics that drives, it's the physiology. And once these cells are connected to their neighbors, they're basically going to keep working on the skin and muscle and everything else that they were doing. And so we did all this in frog. We are now moving, as Juanita showed you yesterday, we're now moving into humans, into mammalian cells and human cell spheroids. So this is some work that she had looking at ion channel drugs as electroceuticals to manipulate the phenotype in glioblastoma cells in vitro. And we're now moving into 3D culture, as she showed you yesterday. Okay, so we've talked about the importance of bioelectricity in maintaining multicellularity and maintaining a collective commitment to morphogenesis instead of unicellular kinds of lifestyles. And so I want to shift now to the role of bioelectrics as a pattern memory. And for that, I want to talk about this organism. So this is a planarian. This is a remarkable organism. Not only are they incredibly regenerative, so you can cut them into pieces. The record is something like 270, I believe, and each piece will restore the full worm. They are also extremely cancer resistant, and they don't age. Okay, the asexual forms are basically immortal, and it's very strange actually, and it took us years to understand why this is happening. Why the animal with the best regenerative capacity, the most cancer resistance, the least susceptibility to aging, is actually the one with the extremely noisy genome, right? Because of their asexual reproduction, they have they're mixoploid. Their cells have different numbers of chromosomes. It's very noisy. So why is that? Well, that's a kind of for a whole other talk. We could do a whole other talk on that. But this is an amazing model system because every piece here has this kind of holographic memory of what the collective looks like, and it can rebuild this. And so what we found was that individual pieces or whole animals have this electrical gradient that tells them one head, one tail. Okay, and so if you amputate this animal, you get one head, one tail. Remarkably, what you can do is you can manipulate that gradient using ion channel drugs, and you can move it to this state that says basically two heads. If you amputate this animal, you will get a two-headed worm. This is not AI or Photoshop. These are real animals. Now, something very important to note is that this voltage map is not a map of this two-headed animal. This voltage map is a map of this perfectly normal looking, so anatomically normal and molecularly normal, meaning that if you look at the markers, anterior markers only on one end, not on the other end, animal. And only when you cut this animal do you realize that it had a different idea of what a normal planarian is going to look like. In other words, the body of a normal, anatomically and molecularly normal planarian can store at least two, probably a lot more, but we've nailed down two, at least two representations of what to do if it gets injured at a future time. That's basically a counterfactual memory. Neuroscientists will recognize this as kind of a very early primitive form of the sort of mental time travel that brainy systems can do when they can recall or anticipate things that are not happening right now. So this is a pattern that will sit there. It's a latent memory, doesn't do anything until the animal gets injured. And when it does, that's when it becomes relevant, because that is the ground truth of what the cells consult when deciding what to build at each end. I keep calling it a memory because if you actually cut these two-headed worms, no more manipulations of any kind, just cutting them in plain water, they will in perpetuity continue to make two-headed worms. That's it. That pattern is permanently changed. We've not touched the genome. There's not been any genomic editing. If you were to genomically sequence these animals, you would be none the wiser that they have a radically different body plan, architecture, behavior. Everything is different. You would not know any of that. And so there is really important information that is stored for very long periods of time, possibly permanently. As far as we can tell, it's permanent, although we do know how to set it backwards, how to set it back to normal now, that is kept in this electrophysiological layer. It is not genetics. The genetics does not tell you whether the Planarian is going to have one head or two. It gives you hardware that settles into a pattern by default that encodes one head, but it's rewritable like any good memory should be. And again, this should not be surprising to anybody in neuroscience. That is what the purpose of the nervous system is to be flexible and to store patterns that were not genetically set in stone at the beginning. Okay, so given that we have the ability of cells to interpret these bioelectrical patterns, because the bioelectrical pattern it's saying build a head here, but the other cells have to interpret it and do all of the the hard work of turning on the various genes and differentiating cells and so on into eyes and brains and so on. Given that cells have the ability to interpret it, can we modify what they're doing? And so this is one of our earliest applications when we used ion channel RNA. And so this is Sherry Au and Vijhoff Pai's work, where they introduced potassium channels into the early embryo in different regions, and were able to tell the cells to build an eye. Okay, and so these eyes have all the right lens, right, and the optic nerve. They have all these things. So note a couple of important features here. First of all, this tells you that the bioelectric signals are instructive. They don't just screw up development or produce errors. They actually communicate organ level information. The other thing is that it's highly modular. In other words, we didn't tell the cells how to build an eye. We didn't talk to the stem cells. We didn't control gene expression. We have no idea how to do any of that. What we found is a very high level subroutine call that communicates the idea that this is where an eye should be. And like any good cognitive system, it can take a very simple stimulus and do all of the downstream processing, right? So hierarchical control to do all of the things needed to implement what it wants to do. We also found something very interesting, that if you section these eyes, you can find that only a few of the cells. So here's a lens sitting out in a tail somewhere in a tadpole. Only a few of these cells were directly injected by us. What they did was instruct their other cells to help, because there wasn't enough of them to build a complete organ. So there's this secondary instruction where we instruct these cells. Once they're committed, once they've bought into what needs to happen here, they will then do things we don't know how to do, which is to instruct all the other cells to participate. And so this is an important part of future medicine. Right now, to my understanding, aside from antibiotics and surgery, we really don't have many, if any, medicines that actually fix anything. We have things that suppress symptoms, but we don't have anything that you take it for a while and then you can stop taking it and it's fixed. The problem is fixed. The idea here is that we absolutely need to reset set points so that the system itself becomes committed to whatever outcome we want, so that we're not trying to micromanage a totally intractable number of moving parts, but actually we get the buy-in of the homeostatic process. And there are many collective intelligences that do something like this, not just cells. I mean, ants and termites, if they find something too heavy to move, they will recruit their nestmates to help them do it. It's kind of a common thing in collective intelligence. So we can make many different organs. We can make ectopic. We've made ectopic limbs, ectopic hearts, with this way by ion channel misexpression, otocysts, ectopic brains. Even, and we'll get back to this in a minute, structures that don't actually belong on a tadpole, such as these kind of fin-like structures. But we'll get to that. And so the idea of this now that we're starting to understand what aspects of the bioelectric properties are controlling downstream outcomes, now we can start to get really specific, and we can ask, okay, can we induce repair of things that go wrong? So here is this is a set of papers by Pai and a few with the modeling by Alexis, where we were looking at the frog brain, and you can see here the forebrain, midbrain, and hindbrain, and various teratogens, nicotine, alcohol, lots of other things, produce characteristic defects. So here you can see the brain is kind of damaged. The eyes are connected to the brain; they're not at the right distance, and so on. And so we asked the question, what's actually going wrong here when this happens? And Alexis produced this computational model that uses the voltage imaging that we performed to understand what happens to set the correct or the incorrect shape and size of the brain. And it turned out that once you have a model like this, what you can do is you can run it backwards and ask it questions, and you can say, okay, given that what's happening is incorrect, how do I bring it back to the correct state? In other words, what channel do I open or close? So those are our control knobs. What channel can I open or close to get the pattern back to normal? And in particular, for a range of defects of the brain, face, heart, and gut, what we learned is that there's this amazing thing called HCN2. And what's really cool about HCN2 is that it's basically like a sharpen filter in Photoshop. If you turn on HCN2, it takes fuzzy or diffuse voltage patterns and it sharpens them so that the boundaries between regions of different voltage are stronger. In other words, cells that are slightly polarized stay where they are. Cells that are depolarized stay where they are. Cells that are slightly polarized become very polarized. And as a result, you sharpen up all the gradients. And that is enough to, when you do that, that is enough to take tadpoles that have extremely severe brain defects. So this is a notch mutation, very important neurogenesis gene. These animals have no forebrain. The remaining tissue is basically a bubble filled with water. They have no behavior. They just lay there doing nothing. You can override this despite the presence of this dominant notch mutation and get a brain that has normal anatomy, normal gene expression, and normal behavior. Their learning rates go back to normal by managing this, by managing the bioelectric pattern. And all we had to do in this case was open HCN2. We did not have to tattoo it in specific cells. We didn't have to manage the fine structure of where it's expressed and how and when. Just that sharpening filter alone was sufficient to override notch mutation and actually a bunch of chemical teratogens as well. Okay, so that's one way to start to computationally predict interventions, is by picking electroceuticals like this using a computational model of what the bioelectrics are doing. We also have a program in appendage regeneration. So some of Kelly Cheng's work using very simple triggers. Again, I'm showing you this because this again reminds us that very simple triggers in bioelectrics, very simple triggers can give rise to very complex outcomes. So in this case, a 1 hour stimulation with a sodium ionophore, which drives sodium into the wound, kickstarts at this non-regenerative phase of life, kickstarts regeneration, even though a wound epithelium, a normally non-permissive wound epithelium has already formed. We took this into frogs, and so frogs, unlike salamanders, do not regenerate their legs. And so what Kelly showed is that after 24 hours of stimulation compared to what normal frogs do at this point, you get immediate turn on of pro-regenerative genes such as MSX1. By 45 days, you've already got some toes and a toenail. Amazingly, the path that the leg takes to regeneration is not the normal path by which frog legs form. It's quite different, but it gets to the same endpoint because you can see eventually you get a very respectable leg that is touch sensitive and motile. But like many problem-solving systems, it doesn't necessarily follow the same path that it did before. It finds a new path to get to where it's going. The most recent work from Celia and Narosha had to do with very large adult frogs, which have never been shown to be able to regenerate anything like this. Again, using a bioreactor, we were able to induce very significant regeneration. But maybe the most amazing thing about it is that we only interacted with the wound for 24 hours. So it's a wearable bioreactor. 24 hours of treatment gives you 18 months of growth, during which time we don't touch it at all. So this underlies the same thing. The amazing thing about, or one amazing thing about cognitive systems is that when you communicate to them, you are not in charge of all the molecular complexity that it takes to make changes in their body. In other words, when I'm talking to you now through this very thin language interface, right, the kind of a low bandwidth connection, I don't need to worry about tweaking all of your synaptic proteins so that you understand and remember what you're hearing. You will do all of that yourself. The whole electric network of your brain and body is basically organized so as to take these low information content stimuli and do all of the downstream steps that it takes, move all the biochemicals around so that the information sticks. And so that's what's happening here. We did not have to micromanage any of this. 24-hour stimulus to convince the cells that you're going to go down the leg building path, not the scarring path. And that's it. After that, we take our hands off the wheel. And that's really what we're looking for. And at this point, I have to make a disclosure because Dave Kaplan and I have this a spinoff called Morphoceuticals, and we're now moving this into this kind of approach into mammals, where we build various kinds of bioreactors that deliver payload and allow an aqueous, tightly controlled environment around the wound where all the ion currents can flow and the cells can feel that they're in a protected, almost amniotic like environment. And then the payload is designed to these are drugs of ion channel drugs and some other things designed to tip the whole system towards regeneration and away from scarring. And so the point of all these examples is that the traditional story that everybody's taught, which is that the cell is the hardware and the DNA is the software, is actually stood upside down by the work in bioelectricity, which reminds us that actually the DNA is what sets the hardware. The DNA tells the cell what kind of channels and pumps and gap junctions and things it gets to have. But after that excitable medium of cells expressing these things has some very interesting and very important properties that allow self-organization of patterns, memory, computation, and all the things that neuroscientists are used to, which means that much like in computer science, where we know that this simple flip-flop can store two different kinds of information, so a 1 or a 0, but not by changing the parts around, simply by temporarily giving a stimulus that changes the way that energy flows through the system, and it can acquire different states and carry different information one way or the other. None of that is visible on an omics approach, right? You could take an X-ray of the system, you could count all the resistors and transistors, you would not know what the information is. And so that reminds us that biochemical omics does not tell the whole story, and we're now applying lots of really useful tools from connectionist cognitive science to try to understand how networks of cells can store memories as attractors in physiological space, but also how it can do things like pattern completion and generalization, because the different layers in these systems generalize to larger scale features. So while you might have chemical gradients at one level, eventually you'll have tissue geometry, axial polarity, organ identity, and things like face patterns and so on. And so the dream here is to be able to have a full stack computational model that goes from the molecular networks that tell you what channels and pumps you have, meaning the targets of your intervention or all these things, all the way through tissue level models that show how it is that these voltage patterns arise, what their properties are in terms of injury and modification, and eventually into algorithmic models. This comes from Slack's book in 1981. So already it was quite clear. Nobody knew how to get to it, but it was quite clear that what we would need to do, it's a algorithmic model of Planaria and regeneration, trying to understand what are the steps that it requires to have a proper worm from a little fragment. But this is the kind of thing that's easy to intervene in. This is actually very difficult, right? Because it's very hard to do the inverse problem of knowing what do I do here in order to get a specific outcome. So this is what we're working on now, is kind of a full stack integration of these different layers, so that we can we can start to infer interventions and birth defects, regeneration, and cancer. So I think that one of the most exciting things about bioelectricity is it's not just another piece of physics that needs to be managed the way that biomechanics is, and so on. It's a communication interface, and it's a communication interface to the very root of the deep problem here, which is the boundary between self and world. When I showed you that embryo that self-organizes out of a huge number of cells, and you cut it into pieces, or don't, every cell has other cells as neighbors, and that whole collective has to figure out where do I end and where does the outside world begin? What are the states that I'm going to work hard to maintain? Right? Are they large states, small states, and in what space? Right? In anatomical space or physiological space, and so on. So bioelectricity is this really critical integrating layer that controls a lot of downstream things, because it provides this multi-scale competency architecture. It aligns the components, which are by themselves active, matter, and smart. It aligns them towards higher level goals. And so I think what we're going to have, in complement to the kinds of bottom-up systems that are exploited today by medicine, we're going to have some very interesting top-down interventions that have to do with the use of electroceuticals, which are, I think, a kind of a subset of a broader field that will also encompass biomechanics and biophotons and biochemical signals, of course, and all of that stuff. But in order to not to micromanage the molecular pathways, but to convey new information, new set points, and alter the boundaries of the systems that are making decisions about what they're going to do. Now, what I'd like to do in the remaining couple minutes is to kind of move past everything that I've told you up until now was about how good these systems are at regaining their normal species-specific target morphology. In other words, they start, even conventional embryos, never mind Planaria, but conventional embryos, they rebuild the entire body from one cell. I mean, that's an amazing, standard development is an amazing example of regeneration, where you restore the entire body from one egg cell. So in all of those cases, whether interfered with in the meantime by scientists or not, it's very good at creating and reaching that correct target morphology. But let's just look at a couple of things here regarding to the question of plasticity. What else can it make? Okay, so beyond repair, let's look at what the possibilities are for bioengineering. Well, the first thing we know is that, yeah, Planaria are very good at restoring their bodies, but actually, if you but the control is not just about the number of heads. If you manipulate the bioelectrical decisions during that regeneration process, you can actually make the same hardware, meaning the same genome with no genomic editing, you can make them visit other attractors in the anatomical space belonging to other species. So for example, this guy with a triangular head can be made to have a flat head like a P. felina, or a round head like an S. mediterranea. This is about 100 to 150 million years of evolution. Also, the brains and the stem cell distribution in the brains become just like these other species. So the same hardware can be pushed into the attractors belonging to other species. So that's weird enough. But what about things that have never existed before? Because one thing you could say is that, well, these attractors are evolution created them, right? It shaped other species to be able to do this, and so this is what we're finding. We're finding other evolutionarily stable strategies that have been rewarded and then selected for in the past. So I want to show you a different kind of technology that I think is going to be really important for biomedical interventions. And when you look at this thing, you might guess that this is some kind of primitive organism that we got from the bottom of a pond somewhere. And if I asked you to guess what kind of genome it has, you might guess that it has a genome similar to some of these very kind of primitive organisms. If you were to sequence it, you would find that this is 100% Homo sapiens genome. Okay, so these are constructs that we make from cells that derive from adult human patient tracheal epithelia. We call them Anthrobots, and what they do is they self-assemble into this amazing little self-motile proto-organism. There's a particular protocol that we use. This is what they look like. The reason that thing was swimming around and moving is because they're covered by little hairs called cilia. So these things beat, and normally in your airway they're moving the mucus around and so on. But here they're able to row against the water. This is a human version of the Xenobots that we made some years back. And there's a few really interesting features about this. First of all, there's never been any xenobots, any anthrobots in history. They don't look like a stage of human development. There's never been selection to be a good anthrobot. So this is a novel construct. One of the things that they do is, if you look at the transcriptomics that compares what genes do the anthrobots express versus normal versus the cells that they come from, about half the genome, about 9,000 genes, are altered in their significantly altered in their expression. Okay, an enormous amount of transcriptional change. They've not been genetically edited. There's nothing wrong with their genomes. There are no scaffolds. There are no nanomaterials, no weird drugs that we're using. They have a different lifestyle. They are freely motile. They live in the same kind of environment physiologically as they did before, except that they're not locked in by other cells. So they're sort of free to explore their new multicellularity. And they take full advantage of it by transcribing their genomes very differently. They also have 4 different behaviors, and we can draw an ethogram of the transition probabilities between like you would for any animal. And they have some interesting features. First of all, if you plate a dish of human neurons, so these are iPS-derived human neurons, and you put a big scratch through it, so you take a scalpel and you put a big wound in, what you find is that they kind of traverse the wound, but then they settle, and when they settle, they can form into something we call a superbot cluster. So basically, there's a bunch of them, and what they'll do over the next 4 days, they'll sit there and they'll reconnect the 2 sides of the wound. Here you can see, when you lift them up, you can see this is what they were doing. They were healing the gap. Who would have thought that your tracheal epithelial cells that sit there quietly, dealing with mucus and particles in the air, have the ability to self-organize into a motile little creature. They live for about 5 to 9 weeks, and they have all kinds of capabilities. This is just the first thing we tried. Okay, this is the absolute first thing we tried was this. So probably they can do many other things that we haven't figured out yet. And the opportunity here, is to use them as a kind of agential implant, in the sense that the drugs and I mean, now we're getting into some smart technology that we're implanting into people, but mostly drugs are pretty dumb. They just they have a target, and ideally they bind it, and that's that. But you can imagine something like this made from your own cells, so you don't need immunosuppression. It has 1 billion plus years of shared history with you, so these cells know what cancer is. They know what inflammation is, what stress is. You could imagine a million applications of using things like this to do helpful things in the body that we actually have no idea how to micromanage. And so I suspect, and I didn't even get to talk about this, we have a lot of things going on around self-training and tissue training and so on. My suspicion is that future medicine is going to look more like a kind of somatic psychiatry than it is like chemistry, because we're going to have to really pay careful attention to what it is that cells and tissues, what inputs they've received, what memories they form, what set points they are pursuing, what are they capable of pursuing, what their problem-solving competencies are. A huge chunk of that is mediated by bioelectricity, and so I think that we have this amazing capability now in this field in particular to address some of this. And so I'm just going to say, sorry, losing my voice. I'm just going to say that now there's some great tools coming online, in particular some AI tools and things that we're doing around creating systems to actually talk using natural language, to talk to gene regulatory networks, to talk to cells and tissues, to talk to organs, and using AI as a translator module to the different layers of information in the body that normally are linked by bioelectricity at different scales. And so I'll stop here and just summarize the main points. I think that definitive regenerative medicine is going to require improved methods for communication and collaboration with the agential material of life, to recognize that we're not dealing with passive matter anymore, that we can really take advantage of some of the competencies. And for that reason, I think the relevance of bioelectricity is not just as another piece of the mechanics. It's actually a component of cognitive science and computer science that can let us reach new outcomes that we couldn't do otherwise. I think that bioelectricity is an amazingly powerful interface to 2 classes of things. First, rewritable goal states, and second, the borders of the decision-making agents. Right, it's the cognitive glue that makes individual cells into larger scale computational units. And in addition to writing these goal states, I think we can also use bioelectrics to learn a lot from the cells themselves as to how they're solving different problems. We have many examples that I haven't shown you today about how cells themselves react to and solve a bunch of problems that we actually have no idea how to do. So hopefully we can we can learn from them by using this interface. And all of the things that I've shown you about the Anthrobots and the Xenobots and some others, we have some other things like that coming soon. All of those things are poised to take advantage and to benefit from everything that's being developed in this field, and all the amazing advances in bioelectricity, because they allow us to now start to probe not just the stability and the robustness of life towards standard target morphologies, but actually towards the plasticity to completely new things that have never existed before, and to use that as a sandbox for really complete control over growth and form in that anatomical compiler that I mentioned at the very beginning. So, okay, I'm going to stop here. I'm going to thank the amazing people who did all of the work that I showed you today. So here are all the postdocs, grad students, and staff scientists who did all of the things that I told you about. We have incredible technical support, many very valuable collaborators that work with us on all of these things. We've had different kinds of funding over the years. 3 disclosures that I have to do. So morphoceuticals, AL, and fauna systems. And of course, the most important thing to thank is the model systems, because they do really all the heavy lifting, and they're the ones who teach us all about this stuff. So thank you so much.